The overall goal of this procedure is to quantify one aspect of the social behavior of flies, their preferred social distance. This is accomplished by first assembling the social space chamber. Meanwhile, a group of flies is habituated to the environmentally controlled assay room by being transferred into a new vial there. After 2 hours, the group of flies is transferred to the social space chamber. They are tapped down and then positioned vertically with a clamp stand. Once the flies settle, their positions are documented, and the distances between the flies is calculated from this photograph by software. Ultimately, the social space assay quantifies a form of social behavior which can be used to study the influential environmental factors and neurogenetics of this behavior.
The main advantage of this technique to quantify social behavior over existing methods like computer-assisted video tracking is that it does not require sophisticated tools or methods, nor large investments in time and training.
This method can help answer key questions in the social behavior field. We have used this method as a diagnostic tool to study the role of orthologous genes thought to be involved in social behavior in other organisms, such as candidate genes for autism spectrum disorder in humans.
I first had the idea for this method when I noticed at UCLA that in our stockroom, flies in their vials did not have a random distribution, but formed aggregates with repeated intervals in between them.
This procedure describes the assembly of the social space chambers. To begin, while wearing gloves, place a triangular spacer on a square pane, aligning their corners in one location.